So again, this sample over here that I've used for the stratified squamous, keratinized one, the most intact one, cells are all still stuck together. They almost look live, except that where there was a nuclei, there's kind of a space there because all these cells are dead, right? They remain stuck to each other and everything, but they're just bags of protein actually. All right, so that's your stratum corneum there. On this sample, it looks a little bit different. You can't really make out the cells anymore, but you could definitely tell it from anything below it right over there. It's starting to flake off over here. And then for this thin skin sample, it's the most degraded for the most part, most flaking off, right? There's still some kind of attachment, but this is that stratum corneum versus the underlying living layers right over there. Yeah, the whole thing is a stratified squamous. Remember, this is just that layer, that specific layer of the epidermis. And then for your stratum basal, any of these, right, would be that layer that is directly connected to the underlying papillary layer right here, right? So you can tell first off where it goes from epithelial to a connective tissue layer over here or over here. Right? This would be the stratum basal right over here, directly connected here, or right, it's not this part, by the way. Right? This is just different distinct layers of these, the epidermis. I didn't go over these, and you don't have to identify them, but it's going to change from epithelial layer to connective tissue under it. Okay, and then this would be the stratum basal. All right, and that's these. I believe that's all those, right? And so next is the dermis. And so the dermis directly connected to the epithelial layer is going to be that areolar tissue. It's going to be the papillary layer. These structures right here are the dermal papilla, right? Over here. And if this is loose connective tissue, then all the cells within it are going to be fibroblasts, right? Or if it was dense, whatever. If it's connective tissue proper, then it is fibroblasts, right? And then all the stuff around it is going to be the collagen fibers. Okay. So the other thing you might notice here, all this other spaces and stuff, these are all like blood vessels and stuff going through it, right? Because this is, heavily vascularized area. This is where like oxygen and stuff is diffusing out of here to the overlying epidermis. Almost any skin slide is going to have the collagen, right? Here's the loose connective tissue with the kind of loosely organized, thinner, more like fine collagen fibers. And then as you move down into the reticular layer, you can start to see those bundles of collagen fibers. And so as you move down into the dermis right here, you could start to see these bigger, notice that over here, like the blood vessels are much smaller and they get a little bit bigger as you go down here because this thick layer of all these collagen here is going to provide a little bit more support. Then you get down here, even bigger blood vessels going through here, right? You could also see all these glands, but you could see here the dermis, and then this gets all this glandular structures over here.
So are the main differences between the papillary and reticular, like the form of the connective tissues?
Yeah, it's a location, number one, right? And the two types of connective tissue, loose, where you have a lot more, because it's more loosely arranged, you'll have more ground substance. So this stuff can diffuse through right here. And then, it's kind of, you could think of it like, these are like individual hairs of collagen, whereas down here is like ponytails or something, right? Big bundles of collagen fibers. And then remember, it's irregular. So that's why it looks like a mess down here. All the collagen fibers are running in different directions. So just like, and the other thing that makes it look like a mess is that all these spaces in here are probably like artifacts, that came during the processing of this sample. So, that's another confusing aspect. But anywhere in this connective tissue, remember, fibroblasts are little dots right here, and then collagen or elastin, right? And the only ones you could see elastin in over here, there's a lot of elastin in here. You can't see it unless you use that other stain, right? You click on number 2 page right here, and then the elastic fibers start to show out. Right? So these little thin black fibers over here, right, are the elastic fibers. That's what's giving your skin when you pull it up, right? You can imagine these being pulled up. You let it go and they kind of stretch back because they're like bungee cords or rubber bands or something. You're giving your skin its elasticity. Whereas your collagen is a little more like leather, right? It's giving it its toughness, right? Resistance against pull. Right? So any of the skin slides, those are the most distinct ones. Like for this one, it's a little harder to make out the difference. Or, actually, to me, it's a little easier with the black staining if you have the red part here, it's a little bit, the dividing line isn't so distinct, but whatever's touching, the top kind of layer that's in contact with the epithelial or closer to it is going to be that papillary layer. And then you have the structures within it is the dermal papilla. Right? So dermal papilla. If it's not really clear, I don't use it. So I like to use, and plus if I do something like this, right? I mean, if I have a, if I draw a little line right over here, and I say, what's this layer? What are you going to say? You're not going to, well, there's a little bit of maybe reticular layer up here, but mostly what's this layer at A? What's this layer at B? Right? What is this layer called? Right? And what's the tissue, right? So the layer is the papillary layer because you're talking about the skin, and it is areolar or loose connective tissue. All right, what's this layer down here? It's the reticular layer, and it's made up of dense, irregular connective tissue. Okay, what are the cells in here? They are fibroblasts, right? What is this red stuff in here? Fibers, those are collagen. And if I have the black stained one, then you could also identify the elastic fibers. So is dermal papilla anything? And then we pretty much went over that, and you just kind of get used to looking at that. It's not too difficult for any of these slides right here. And then I gave you, the hypodermis. That again is just the same as identifying any kind of the adipose tissue, right? So in this sample, right, this up here is the epithelial layer. It's in purple. You get down here, this layer, is it super distinct between loose and dense? Not so much, right? But you can kind of tell it goes from it. You can kind of see the textural differences. And then here you start to get all these hair follicles and glands and stuff like that. But when you get to these bubbles down over here, that's the adipose tissue. So that's your subcutaneous layer. Right? These are the fat cells. That's subcutaneous layer or hypodermis. So that is your skin basically. And most of it, we covered for epithelial or connective tissue already, right? You just want to, the only new stuff is that this is called the papillary layer. This is called the reticular layer of the dermis. And then what's the top layer? The stratum corneum. What's the bottom layer? The stratum basale. Lecture-wise, there's just a little bit of stuff on the stuff in between it right here.